Greetings, welcome to the 17th H3Africa Consortium Meeting. My name is Sharley Aloyo, and I'm part of the research team at the Integrated Biorepository of H3Africa, Uganda. So I'll be presenting an abstract entitled Prevalence of linezolid resistance isolates in a collection of MDR and XDR Mycobacterium tuberculosis isolates from Uganda. Drug-resistant TB continues to be a public health threat. And in 2018, WHO recorded 500,000 new cases of rifampicin-resistant TB. And out of these, 78% were multi-drug resistant. And also, WHO demonstrates that globally, the number of individuals seeking treatment for MDR TB between 2015 and 2019 demonstrate an incremental graph as seen here in this slide. Linezolid has been recommended for treatment of MDR or XDR TB. This is because it has demonstrated clinical and radiographic improvements when included in treatment regimens. Linezolid acts by inhibiting protein synthesis when it binds to a region in the 50S ribosomal subunit, thereby cutting short the process of translation. However, cases of resistance against linezolid have been reported, and this is basically caused by mutations in genes that encode the binding site and then proteins associated to this site, which is the L3 protein. Problem statement, tuberculosis treatment success is approximately 80% for susceptible strains, but this success decreases to 30% when strains are extensively resistant. This poses a big threat because of the increasing emergence of MDR and XDR TB across the globe, as demonstrated in the slide 1 by the bar graph. And there is also limited data on the phenotypic and genotypic correlates of linezolid resistance in Mycobacterium tuberculosis, especially in Uganda. So this research comes in to bridge this gap. The objective is to determine prevalence of phenotypic and genotypic linezolid resistance correlates in MDR and XDR Mycobacterium tuberculosis isolates from Uganda. Methodology, of course, after getting an IRB approval, phenotypic DST will be performed on isolates in the BS cell 3 lab where TB is cultured. And in the isolates that demonstrate linezolid resistance phenotypes will then be transferred to the molecular lab where DNA will be extracted, and then PCR will be performed to amplify the genes that encode this resistance mechanism. Then information, of course, will be analyzed and then presented or disseminated to show the occurrence of these resistance patterns in the population of isolates that we have. Then going forward, the next steps would be to perform prospective sampling to gather more information on the occurrence of these resistance correlates in the population. So it will more be like sampling from different regions to get a map of the occurrence of these resistance patterns in the population of Uganda. So this basically brings me to the end of the presentation. Thank you very much for listening. May you enjoy the rest of the event. Thank you.